Hi, good morning, everyone. Thanks for the organizers for this really nice workshop. And I'll tell you a little bit about how we try to use extreme value statistics in analysis of data. And I work on bacterial physiology. Okay, I work on bacterial physiology. It's coming up on 100 years on this landmark book over here by Enrici. And I'm always, it's really fun to see how people were interested in this so long ago, and they were looking under a microscope and drawing it even by hand. And so, yeah, so this has been a topic of research for many, many years, how bacteria grow, how they divide, et cetera, et cetera. And now with modern technology, well, before modern technology, the point I want to make first is, even when Enrici plotted these cells by hand, it was very clear that they're different from one another. There's variability between single cells. But even if we knew everything about single cells, it would be a very long stretch to understand the population. And this is quite different from what we're used to in statistical mechanics, where we know the properties and the statistics of the single element, and from that we learn the properties of the population. But in a cell population, things are quite different. First of all, it's a dynamical system. The variability itself is dynamic in many, many properties that we can measure. Any property that we can measure is variable, and it also changes across time. Cells grow and divide. They create lineages. These lineages create memory. They inherit their properties to the next generation. And this creates, of course, correlations inside the population. And moreover, every cell is really an open system, right? It's open to its environment. It exchanges substance with the environment. It's sensitive to spatial gradients and other features. And because of that, the individuals in the ensemble are not independent. Through the environment, they can interact with one another directly and indirectly. So I think many of the people here, or some of them, have been interested in this question of how to relate single cell properties to a population, a growing population properties. I've been interested in particularly in two main aspects of this. Sorry, before the aspects, I want to say this field is both driven and also constrained by technology. So we've had several breakthroughs, for example, the microfluidics devices that people now use, which enable to follow single cells, and that extended the time span tremendously. You previously could not follow single cells for so many generations as you can now, because if you are in a population, of course, it saturates very quickly. But that has the disadvantage that each lineage is separated from the other ones. A little bit of that was another step forward was this sisters machine that was developed and allowed us to look at two sisters after they divide and look at the correlations and the memory. So that's already a tiny bit starting to look at how things develop in a population. But still, they're basically, for a very long time, they become independent. Every pair of sisters are still independent. And also, another technology that has been developed is the droplet technology. So here you have small droplets, or there are different kinds of droplets, from microfluidic to millifluidic droplets that allow you to have a small population in a shared environment. And that can actually give you access to interactions between an intermediate number of cells, at least. So, right, so we've been interested in two types of general classes of problems. The first one is the statistical signatures of interactions that I'll be mainly talking about today. Another aspect is the quantifying of ergodicity in these populations, or in other words, the statistical significance of different lineages. But I, of course, won't have time to talk about everything. I want to focus on the droplets. And what I'll tell you about today is a product of a collaboration, a Human Frontiers collaboration from a few years back, where really the bulk of the effort there, we spent an enormous amount of time on building the machine. This was in collaboration with a company called Millidrop. And really, a lot of effort went into understanding what's going on, how to build them, how to make them uniform, how to make them repeatable, the droplets. But ultimately, I think we ended up understanding something about the cells themselves. I hope so. This is our experimental system. I won't go into the technical details too much. Yeah, this is not working at all. I don't know. Yeah, maybe. I want to point with the pointer. Oh, that's the pointer is working. Fine, I'll do with the pointer. Okay, so this is thank you. This is the tube. So basically, the way the machine works is there's a tube. It sips medium, it sips cells, and then it places the cells inside droplets. These are the droplets. They're about 0.5 millimeter cubed in this experiment. And these are air spacers. You have to put spacers so that they don't merge, so that they have a long lifetime. This is a scale of millimeters. And in this particular experiment, we grew Pseudomonas fluorescens, which is a bacterium of similar size to E. coli. But the nice thing, one nice thing about Pseudomonas is that it fluoresces spontaneously. It has a fluorescent signal, and then this fluorescence can be calibrated against a concentration of biomass and can be calibrated against cell number. So we have a good handle on cell number inside each droplet. And if you now put few cells in each droplet, this is what the raw data looks like. Okay, so you have approximately, this allows you to monitor, I think, something like 3 orders of magnitude in cell number per droplet. This is per milliliter, but per droplets, it's between 1,000 and 1,000,000 cells for each droplet. And this is what the raw data looks like. There's a blind region because of the detection. That's a technical point. And these are the growth curves that we see in the droplets. Okay, so they're one on top of the other. And we have a lot of statistics. So we do preserve the high throughput kind of nature of the microfluidics because we have many repetitions. Okay, so what can we learn about the cell population growth? So the coarse-grained parameters that I can take out from this growth curves, first of all, is the growth rate. In the intermediate range where the growth is exponential, I can extract the growth rate. Then I can extract a capacity or a maximal population size in the droplet. And then there's the lag time over here, and that's maybe the most interesting parameter. Yeah, so these two parameters, growth rate and maximal population size, of course, depend on the type of bacteria, on the medium composition, etc. But the lag time is the more complex one. Because I'll talk more about that later, but that depends really on a lot of detail and history of the problem. So we extract the parameters. I just want to say there's been a lot of work on, you can generate fancy algorithms to get out the growth rate. And here, but everything that I'm going to tell you is not dependent on that. These data are clean enough. You can just extrapolate back the initial population size and then get a growth rate and the time, the lag time. So these gross parameters, these coarse-grained are quite robust in the data, and they don't depend on a very complicated analysis that we do. Okay, so the statistical power of this system comes from the fact that you can control the inoculum size. Okay, you can inoculate each droplet by a well-defined number of cells between 1 and 1,000 here, you can see. And then you can repeat that. You can have an ensemble of those, which gives you like several hundred repetitions for each inoculum size. So the parameters we can test as a function of the inoculum size. The growth rate is quite stable. The variance decreases a little bit, but that's not a huge effect. The capacity also is very stable. And what is dramatically dependent on the inoculum size is the lag time. Yeah, they were taken out of the freezer. They were taken out. So, okay, so everybody who's familiar with this knows that lag times are extremely sensitive to everything. And basically, this effect has been known. This is what's known in microbiology as the inoculum effect, or one manifestation of the inoculum effect. And that, in a sense, it says that if you have fewer cells, it takes them longer to start growing. And that's a really good point, because this is not a universal effect. It happens when you take them out of the freezer. It happens when you take them after starvation, when you take them after exposure to some stress. And people have worked on this, and it's very sensitive to all these details. So Oded has worked on this problem, or a related problem, with Nathalie Balaban, and they found that timescales are dependent on the history, on the amount of time in stress. These guys also found that the duration of starvation changes the mean lag time in a kind of complicated way. This paper studied the effect of antibiotics exposure and the history of antibiotics exposure. So this is a very delicate effect. It depends on a lot of things. Yeah, question? Okay. Well, it's never zero, but it's smaller. That's actually another point. It depends. Another thing that it depends on is where you take it from. If you take it from exponential or from stationary phase, that makes a difference. So the dramatic effect that people have reported is always after, or taken from deep freeze, or after some. So it depends, of course, on the state of the culture before. Yeah. What do you mean? No. It doesn't. It's how you sip the cell. You don't put them one by one. It's the concentration. So it's the same time. Yeah, it's the same time. And you'll see in a minute the time scales that are relevant here. You'll see that even if there's a tiny difference, it's not in the same scale. Other questions? So this. Oh, sorry. Yeah. Okay, that's the topic of this talk. So, despite the fact, I'll just say that despite the fact that this has been reported, I think from the first time that a lag time has been reported, people have noticed that there's dependencies and sensitivities, but there has not been a well-known. It's been debatable for many, many years. Yeah, another question. So that's a really good question, but in this experiment, the volumes are constant. So in this experiment, I think it's the same. These are historical data. I'm not going to compare quantitatively to these experiments because every one of them has different features. All of this only comes to illustrate the fact that this effect is really sensitive. It's sensitive to the way the experiment is performed, to the way the culture is taken before the experiment, to various things that, and this is, I think, part of the reason why it does not it has not received like a well-accepted explanation that is relevant to all cases. Because, you know, it's really, I don't know how to call it, it's sensitive. Okay, so, right, so what can we learn, what does this, our experiment, what new things our experiment contributes is, let me just give you an overview of what the statistical data looks like, and then we can see what we can learn from it. So what we can do, these are three repetitions of the entire experiment. Like I said, we have several hundred repetition or droplets for each condition. So we can do the average time. This is the classic inoculum effect. Average time decreases as a function of inoculum size. We can also compute the variance, and the variance also decreases. So this is a log-log plot, and you can be impressed, at least qualitatively, that this decays slower than a power law. And we'll get back to that later. So both of these first moments decay, but they do that in a coordinated manner, and they retain a variance to mean kind of quadratic relationship across the inoculum sizes. And of course, these are just the first moments, and we have the probability densities here and the cumulative distributions for the whole range of inoculum sizes. And so this is quite quantitative data, and we can see whether the goal is to see, can we explain something about the source of this effect? Okay, so what is the origin of the effect? So one classic verbal explanation that's been around and makes sense in some way, it's logical, is the following. If there's a variability among individual cells in the lag time, now if we have a larger inoculum, we are sampling further into the tails of the distribution, we are getting faster tails into the initial condition and faster times into the initial conditions, and that'll give us a more rapid increase. And here, this of course encompasses the simple way to translate single cells into population. So you just sample more, and then you get faster times. So in our experiment, we have measured the single cell lag time distribution. It's approximately a log normal distribution. You can see now the time scales. The lag here, average lag is close to 7 hours, and it ranges between 4 and 10 hours. And so a very simple-minded thing I can do is I can take this distribution and sample from the empirical distribution and simulate the droplets on my computer, and then I will see. I take several hundred of drops, I draw initial lags from here, and then I see what will happen as a function of when will I reach, so I have to set this threshold like I have in the experiment. So when will I reach the threshold? Okay, so this is the experiment, and this is the simulation that takes out of this distribution. And clearly you can see that this variability cannot explain this decrease in average time. So this is the simulation, this is the variance, and this is the mean. The variance is actually increasing very rapidly, but the mean is almost not decreasing at all. You can do a very simple back of the envelope approximation here and see that by the time, I don't want to go into it because it's not so important, but by the time these, if these are the fast cells and these are the average cells, by the time these start to grow, these guys have not made up a substantive population inside the droplet. But on the other hand, there's averaging here. So the main effect here is of averaging. Yeah. All right, let me just, let me first highlight what this is exactly, how this is measured. Maybe that'll answer. So this is measured from, I don't know, like 300 or 400 different droplets, in each of which you put just one cell. Okay, I put one cell as an initial condition, and then I measure the lag time. But one of, the drawback of the system is that you're not following them one by one after you've inoculated them in the droplet. You can only follow the population side or the micropopulation size. So I can't tell you exactly what's going on inside there, but I can tell you that this is the single cell inoculum, okay? I think we'll clear it up a little bit, but that's a good point because really, these technologies have advantages and disadvantages. I do not follow lineages. I do not follow single cells as they go along. I only have yeah. What do I mean here by mean lab time? Okay. Let me say again what I did here. So I take this as the baseline of the single cell, and now I simulate the following. I take droplets with one drawn sample from this, and then I take droplets with 10 independently drawn from that distribution, and then I take droplets with 100 independently drawn. So I'm okay. This distribution is measured in droplets that have started from a single cell. Yeah, I have enough of them. Okay, is that more questions? Yeah, but not enough. Of course, but not enough. So wait, let me just add something, a detail to the simulation. So you can ask, for example, maybe there's a correlation between short lag time and fast growth rate, right? That would have to be an enormous effect, dramatic effect. We don't see that, but that even if you do put a correlation inside, it's not enough. Okay, it's not the timescales, the differences and the timescales involved are not such that you can wait for a sweep or a takeover or something like that. It's not consistent with it. Yeah, more questions. Okay, again, for example, let's take the 64, okay? I simulate 300 times a droplet that starts with 64, and 64 times I draw from this distribution, and then over these 300, I have a mean and a variance, okay? All right, so this is clearly not a good, sorry. Of lag time? I think probably, I think we do. We do have. Oh, okay. And also Natalie has measured this, and she has done a lot of work on measuring lag time. Are you asking if it's the same distribution? That's a good question. Just reminding you that this is, it could be depending on the organism, the microorganism could depend on a lot of things. And I have to say a caveat here. Everything that I'm telling you here, I'm not going to claim that it's always true. So this is just one case when we can handle this, get a handle on it. But that's a really good question. I think we should probably look into that and see how different the distributions are from one setup to the other. That's not, I'm happy understanding something about this. E. coli is not special. So, yeah, but the comparison is certainly of interest. Okay, so this is clearly not what's going on here. So let's go to the other extreme. The other extreme is that this is a very collective effect. And by collective effect, I mean there are strong interactions between the cells. So imagine the following scenario. Suppose the first cell that starts to grow immediately signals to all the other cells, it's time to grow. Everybody starts growing at the same time. Okay? So how would I simulate this situation? Again, I start from the single cell distribution of lag times. Again, I make all these, I simulate 300 droplets start with the same number. And among this number, I always choose the smallest lag time in the sample. Okay, I choose the smallest lag time in the sample, and everybody starts to grow at that time, right? This is what you see, and you see that I don't need to tune anything. The blue in the background of the data are this simulation, and the only thing it depends on is this distribution here. Okay, and this is, of course, an extreme value problem. The other one is the previous simulation that we said was not describing the data well, But now if I turn to this extreme scenario of synchrony, right, everybody wake up at the same time, I'm getting something which is quite compatible with the data. Right, is that clear? Okay, so it's compatible, and it immediately brings me to think about the extreme value framework. I guess I can skip this slide in this audience. Everybody knows about extreme value statistics. So I have three universality classes. The one thing that I want to say here is that while the distributions are universal and depend only on the tails of the parent, the constants that you need to take to normalize, right, the rescaling constant are not universal. They depend on the distributions. Okay, so what if I'm right? If I guess I'm running out of time, so I'm not going to give you the spiel about the intuition of extreme value. Probably everybody knows it here, but the point I want to make is to connect it directly to the data. What does this give us about the data? It gives predictions. So the first prediction is the mean as a function of inoculum size, which is the average minimal value as a function of sample size. That is predicted by extreme value theory. And for the family of distributions to which our parent distribution belongs, which is along normal, actually it's the same universality class as the Gaussian, that is a square of the logarithm. So over here is basically the average and the standard deviation. It's a linear function of the square root of the log of the sample size. All right, this is a first prediction. The second prediction has to do with the variance. The standard deviation is 1 over the same square root. And over here, you see the same data that I showed before from 3 different experiments together with this prediction. Okay, so reasonable agreement. This is, I just want to say, this is not a very standard dependence, right? Square root of log n and 1 over square root, it's kind of... What? No, it's a typo. It's the square root of log. This is the right formula. Sorry, this should be a square root of log of n. Thanks. All right, so there's a first prediction that looks good. Another prediction that comes from extreme value theory has to do with the scaling properties of the distribution. So the distribution you can show, this is the generalized, or the Kavita gave this introduction. This is the generalized form of the extreme value. You can show that for any value of k, if you have a random variable that is distributed according to this with a particular k, a particular shape factor, and you transform, you do a affine transformation of that variable, that is still with the same k. That still goes under the same shape factor. And in particular, you can show, it's easily to convince yourself that if you take the empirical data, you subtract the mean and divide by the standard deviation, you're only left with the shape. And if indeed all of these are taken from extreme value samples, we should get a collapse of the distribution after 2-parameter scaling. Okay, so these are the data original. These are the scale data. This is an asymptotic theory. So if you allow me to take 4 out of that and starting with 16, there's a really very good collapse of the distributions. And actually, there's a black dashed line here, which is an extreme, which is the shape of the distribution taken from the theory. Okay? So that also works quite well. So I've told you about 2 extreme cases, one of them independent cells and one of them completely synchronized cells. You can think about the intermediate case where there's a signaling between the cells, but not all of them, immediately receive the signal. Again, I'm going to cut this a little bit short because I want 2 minutes to say something general about extreme values. So if you do this, you can define 2 variables that compete. If you imagine that the cells are generating some getting out of their, secreting, that's the word, secreting some molecule that the others can detect. Then you have two timescales you need to compare, the variance between different lag times and the signaling timescales. So if you have one of them very much shorter than the other, one extreme, this is the independent cell regime where the variance is much smaller, and anyway, by the time they get the signal, they would have already woken up and started growing anyhow. And the other extreme is where the signal is very fast. Yeah, question? Yes. 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 Yeah. Yeah. You can do it. We did that. That's a control experiment that was done. Oh yeah, there's something in the medium that plays a role in this. All right. So again, I don't want to go into a lot of detail, but you can also simulate this situation in an intermediate case, and you can see that the result of this simulation. So you can set up like a little model where you have secretion and then a threshold, and everybody has to reach the molecule has to reach a threshold for everybody to sense it. And the bottom line is that the experiment is consistent, the simulation is consistent with the experiment in the region of a very low threshold, which means it's in the region of strong interactions, right? So you only get the order magnitude of numbers of the experiment if you assume that you are in the regime, close to the regime of a single liter cell. 2 liter, but small. It's not that you have to wait for many cells to get the signal. Okay, so this is the end of this story, and I want to say like 2 minutes. I want to take 2 minutes just to say something in response to previous lectures that were here, and that's a note on empirical distributions and extreme value theory. So we had a discussion yesterday about whether when you see a distribution which is of this type, right? If you see one of these universal, does that mean that there is an extreme value problem underhand? Does that mean that necessarily there's an extreme value problem? So a while back, we were interested in protein distributions in microbial cells, and we measured these. This is a typical distribution of proteins. You tag the proteins by fluorescence markers, and then you take a large population, in this case it's yeast cells, and then you see a distribution, and this is high quality data. There's a big dynamic range here in probability. There's a big dynamic range in expression, and you try to fit these data. So my first comment is that it's very hard, like it was already mentioned yesterday, it's very hard to tell apart these long-tailed or heavy-tailed distributions from one another when you have a finite data set. For example, in this case, the Frechet distribution and the gamma distribution, you can tune the parameters such that they look almost exactly the same. Okay? And that's something that always happens even if your data set is large, you hit the tails and you run into problems. And that's when we started looking at these scaling properties. So in this experiment on the proteins, we found that a large set of experiments from different microorganisms, different conditions, different proteins, they all scale on one another. By the way, this is the Frechet distribution. Look how beautifully it fits here. But still, I'm not so impressed by that. Maybe it could have been fit by a log normal as well. So this is the scaling property, and this is the relationship between the variance and mean, which is again quadratic. And all of these families of distribution have location, scale, and shape parameters. And if you fix the shape parameters, they always scale with 2-parameter scalings. So, okay, so what does this mean, right? And I was, for years, I didn't know what this meant, and we now have explanations, but I want to highlight one thing. At some point, I started reading all of Tibor's paper and other people's papers, and I started noticing that people see these extreme value distributions in many different physical and natural situations. So the first, I think, paper, or one of the first ones, looked at power fluctuations in turbulent flow, compared that to magnetic global magnetization fluctuations in the XY model, put the distributions one on top of the other. This is a one-page Nature paper, if you don't know it, and said, okay, so look, there's many different things going on. different systems, and they look very similar, quantitatively similar to one another. Then there was the paper about the 1/f noise, which got a similar distribution. Then there was galaxies, and people looked at the fluctuations in some rivers, and on and on. And then, you know, the impression was that there's something very universal about this. And I thought about this for a very long time. I couldn't find a real good answer. It brought me to think about distribution of, you know, is there a limit theorem which is analogous to the central limit theorem for strongly correlated variables? I then realized that this is a topic of much debate in the mathematical literature. So, you know, we converged on different way of looking at it. But I mean, this to me remained an open question. Why is there so much evidence for this type of distributions in nature? So, you know, waving your hands, you can think if you go to systems that are much larger than the correlation length, you go into the central limit theorem. If you are near critical or for any other reason, you are in a regime where the system size and the correlation sizes are comparable, maybe there is something universal there. So I was not able to find an answer to this question. I would be very happy to discuss it with people who know more about these things here. Yeah, so yeah, this is, I guess that's it. Sorry for going over time. So yeah, so the bottom line from this work, I think, is that using this framework to analyze the data taught us something. Of course, biologists don't look at this as a proof. I don't actually either, but it gives you a direction to look at the data and learn maybe something about the underlying biology. Thank you.